Hello, everyone. This is Dr. Lynn Toohey with NutriWest. Thank you so much for tuning in. We have a great audience today, and the audience keeps growing each month that Dr. Dunn has been doing her methylation series. And today we have the fourth in the series, so we're all very excited to see what Dr. Dunn is going to present to us today. Now, if you've missed methylation series 1 through 3, just refer to NutriWest.com. And if you do not have a distributor code, then just call your distributor and get a distributor code to access the website for the webinars. You can also call 1-800-443-3333, which is the home office if you don't know who your distributor is. Again, that's 1-800-443-3333. And without further ado, here is Dr. Dunn.
Thank you, Dr. Toohey. I appreciate your help with all these methylation seminars or webinars. They've been pretty amazing, and I'm learning a lot through this process too, and I hope you guys are learning a lot. This is the fourth in the series. Our first webinar was on basic methylation, sort of the introduction and what it's all about. The second one was on some variants, what was the second one? Anyway, we were just covering different aspects of the methylation pathway and traveling along. So this fourth series we're doing is on the nitric oxide synthase variants and their impact on health. The second one was on the vitamin D receptor, that's what it was, and the third one was on the COMT and MAO variants. So they just get more and more interesting, and like I say, I've learned a lot, and especially on this one, I was just blown away by the information on the nitric oxide synthase pathways, and I hope you get a lot out of this as well. So let's dig in. I'm having technical difficulties. Why is this not going? Okay, hold on. There we go. Okay. So just as a review, if you take a look at these pathways up here, this is where we've talked about in the past. These are our methyl generators that we talked about initially. This is where this is the B12 pathway and the folic acid pathway here, and this is how we generate methyl donors in the form of SAMe. So when we begin the process of creating methyl donors, as we have learned in these seminars, we want to make sure that all these other pathways are functioning and balanced. Otherwise, we can get into some ill effects, basically, when we start to maximize our methylation pathways. So we're going to, this seminar, we're going to be covering this area over here, the nitric oxide synthase pathway, and its effect on creating peroxynitrates and superoxide radicals, free radicals, and oxidative damage. This thing is, okay, there we go. So again, here's the close-up. So if you look at arginine in the production of nitric oxide, ornithine is involved there, ammonia can be produced, but some of the bad guys down here, peroxynitrites and superoxide radicals, which can cause neuronal damage. So we want to look in detail at this whole pathway, the good, bad, and the ugly. So let's first look at the functions of nitric oxide. It plays a role in vasodilation of the blood vessels. It contracts smooth muscle, creates proliferation of smooth muscle, reduces platelet stickiness. It's anti-inflammatory. It's an antioxidant. It's a blood thinner. It's involved in cell signaling. It regulates peristalsis. It's a neurotransmitter. It increases blood flow to the muscles and organs. It stimulates metabolism, increases endurance, reduces atherosclerosis, regulates insulin secretion, regulates airway tone, and it's involved in angiogenesis and neural development. It's pretty amazing. It sounds like, yeah, let's get some nitric oxide going because it's a very positive molecule. Some of the other functions, it relaxes the smooth muscles of the gastrointestinal tract and promotes peristalsis, stimulates the release of gonadotropic releasing hormone from the hypothalamus. It stimulates the release of pancreatic amylase from the pancreas and stimulates the release of adrenaline, that should say from the adrenal medulla. And it declines as we age. This was a study where Japanese analysts have actually discovered that by the time we reach our 70s or 80s, we create and use 75% less nitric oxide than we did when we were in our 20s. So here are some of the factors that go into how we create nitric oxide and how we break it down. And I think, as we've gone along, I've tried to impress upon you that it's all about balance. So too much of a good thing can be a bad thing. So what we have to look at is how do we create nitric oxide, and then how do we break it down before it becomes a peroxynitrite molecule that has free radical damaging qualities. So some of the things we need to look at are the cofactors. Nitric oxide synthase, that's the enzyme that's involved in the production of nitric oxide. It's unusual, and they require 5 cofactors. Flavin adenine dinucleotide, which is created in the Krebs cycle, FMN also, and then the heme molecule, tetrahydrobiopterin, which is created through methylation, and then calmodulin. So let's look at those factors in a little more detail here. So here's just a little breakdown of the nitric oxide pathway. We're looking at where does nitric oxide come from. If you start on the left-hand side there, so we have food and body proteins that create arginine, and that goes down through this pathway through ornithine. In the process, we can create creatine, agmatine, and urea, which is a waste product right here. And as we go around the pathway, here's another enzyme, the OTC enzyme, that's actually on our pathway. And it's involved in the creation of ornithine to citrulline. And then we create nitric oxide down this way. It's involved in the lungs, the blood, and the kidneys, and then it can be excreted out the urine. So there are 3 different types of nitric oxide. We have neuronal, or nitric oxide. The nitric oxide synthase 1 enzyme is creating the neuronal nitric oxide. The I nitric oxide, which is inducible, that's a nitric oxide synthase 2 enzyme that helps create that. And then the endothelial nitric oxide comes from the nitric oxide synthase 3. So there can be variants in these 3 genes, NOS1, NOS2, and NOS3. And as we've been looking at through the methylation pathways where we have genetic variants, then we have difficulty creating the end product. So in this case, if you've got a genetic variant in any of these 3 genes, then you're not going to create nitric oxide efficiently, and you're going to end up with some deficiencies of nitric oxide and creating some cardiovascular issues, some immune issues, and some neurotransmitter issues. So all 3 are products of distinct genes. They're located on different human chromosomes. Kind of interesting here that the same product, nitric oxide, is created by 3 different genes on 3 different chromosomes, and each of them has a characteristic pattern of tissue-specific expression. And all 3 are expressed in the airways, so it has a very strong effect on our lung issues. Like asthma is a classic example of nitric oxide pathway issues. So again, here's the breakdown of the 3 different kinds of nitric oxide. And the neuronal is involved in synaptic plasticity, blood pressure regulation, atypical neurotransmission, and penile erection. So that's the one that we think of, when we take Viagra, it increases nitric oxide, it increases erection in men. The inducible nitric oxide is involved in nonspecific immune defense. So in the presence of bacteria, funguses, viruses, etc., it stimulates the production of the inducible nitric oxide, which helps fight these infections, and it's involved in the mediation of inflammation and involved in septic shock. The eNOS, or the endothelial nitric oxide, is more involved with vasodilation, vasoprotection, and prevention of atherosclerosis. So again, a little more detail. The neuronal nitric oxide acts on the brain and the nervous system, enhances the ability of the brain and the nervous system to adapt to change, communicate between neurons, and recover. And because good brain health is vital to absolutely every organ and system in the body, we want neuronal nitric oxide levels to be sufficient and active. The neuronal isoform is involved in the development of the nervous system. It functions as a retrograde neurotransmitter important in memory and learning. NNOS has other physiological functions, including regulation of cardiac function, peristalsis, and sexual arousal in males and females. And in the heart, it protects against cardiac arrhythmia induced by myocardial infarction. So different ways that it helps boost the memory. Researchers reveal that nitric oxide enhances memory, baffled thinking. I like that. I like the way they said that, baffled thinking, and slow response in 3 ways. It helps deposit memories as they're created. It helps retrieve memories in the future and go back and remember events. And it helps brain cells send, transmit, and receive signals more rapidly. So one of the things, this is when we look at methylation, several of the things that really stand out as kind of a tell when a patient comes in to see you is memory. It's a big thing. A lot of people as they age begin to have issues with word recall and just memory in general, and that's a classic sign that they are probably having difficulties in methylation. So this is one of the ways that we can spot the fact that they might be having an issue maybe in the nitric oxide synthase pathway, so it's a heads up. The eNOS, again, is involved in tissue recovery and regeneration. It enhances blood flow, it dissolves plaques, it dilates blood vessels, it's enhanced by exercise, and it's compromised by autoimmune diseases. So again, just to reiterate, we're looking at the dilation of blood vessels. If blood vessels are constricted, then we're looking at high blood pressure, reduces platelet stickiness. Again, with platelet stickiness, we're looking at thick blood and high blood pressure. Reduces monocyte stickiness, reduces multiplication of smooth muscle cells of the artery wall, and reduces the release of superoxide radicals. We're going to be looking at superoxide radicals in a little more detail, but basically, it's a free radical. It's going around and causing damage throughout all our cells, and especially in the blood vessels, it can cause some major damage. And it reduces the oxidation of LDL cholesterol, which is a major component of plaque. So here's some symptoms of the endothelial nitric oxide system dysfunction. Cold hands and feet. I know we often think of thyroid more often with cold hands and feet, but if their thyroid is good and you feel like they're balanced in that pathway, and they're having hair loss, those two I think of thyroid. But if they're unresponsive to what you're doing with the thyroid, think nitric oxide. Weak fingernails prone to fungal infections are a giveaway. Immune dysfunction or overactivity of the Th1, Th2 systems. Hashimoto's is a classic sign of nitric oxide dysfunction. It can cause leaky gut. So this is there are several areas in the methylation pathway where we see leaky gut. One of them is histamine. One of them is the nitric oxide pathway. And the other one involves the CBS pathway. So lots of areas where you can have leaky gut. And when you start to get this balanced, leaky gut tends to go. And those food sensitivities are involved with that. And inflammation that's involved in that tend to disappear, which is quite exciting. The chronic inflammation, of course, and low brain oxygen or memory issues, which we talked about. So overall, the research shows endothelial nitric oxide plays a big role in preventing and taming autoimmune disease due to its inhibition of overactivity of both the Th1 and Th2 systems. And this was the study out of a couple of prominent journals here. So the inducible nitric oxide is the second kind, and it produces large quantities of nitric oxide upon stimulation in response to lipopolysaccharides, cytokines, or other agents. We're going to look at that in a little more detail here. Nitric oxide has a cytostatic effect on parasitic target cells. The whole reason that our body makes it is to try to kill off the bad guys, but in the process it can create some peroxynitrite molecules that can cause some problems. So inducible nitric oxide synthase contributes to the pathophysiology of inflammatory diseases and septic shock through the activation of NF-kappa B. And maybe you've heard of NF-kappa B or have looked at it in the past, but it's one of the major players in the inflammatory cycle. So when we go after bugs, we create all of this nitric oxide that then can become an pro-inflammatory situation. So again, here, so we have the promoters of NF-kappa B, which are oxidants, lipopolysaccharides, allergens, bacteria, viruses, and cytokines, will turn on the tyrosine kinase activity, which then activates NF-kappa B, which goes through transcription, translation, and assembly, and in response, we make the nitric oxide over here. So this is involved in the host defense, apoptosis, necrosis, the inhibition of cellular enzymes, vasodilatation, and genotoxicity. So it's a mixed bag. It's how we protect ourselves. It's how we create a reaction to invasion or to toxicity or to allergens, but it can also kind of go the wrong way. So nitric oxide, again, is generated by phagocytes, and the inducible nitric oxide kind. It's activated by interferon gamma and tumor necrosis factor. On the other hand, we have some things that will take it down. Transforming growth factor beta provides a strong inhibitory signal to inducible nitric oxide. So it's how we get it back down again. In this way, the immune system may regulate the armamentarium of phagocytes that play a role in inflammation and immune responses. It's amazing. Our body is so amazing. We just have systems for everything. When we get too much of the nitric oxide created, we have this whole system in play to sort of tone it down. Nitric oxide is secreted as free radicals in immune response and is toxic to bacteria and intracellular parasites, including leishmania and malaria. It's a great defense. When NO forms, it has a half-life of only a few seconds. This is the problem. It's there and then it's gone, in large part because the superoxide anion has a high affinity for nitric oxide. So we have both molecules. They have an unpaired electron, making them highly reactive with each other. They really like each other. Therefore, the superoxide anion reduces nitric oxide bioavailability and forms peroxynitrate. There's that ONOO molecule. I call it the oh no, because it's bad news. But basically, what happens is if we've got too much superoxide being created, then it creates an overproduction of peroxynitrites. So we'll look at that again in more detail as we go along. So nitric oxide also avidly binds to hemoglobin in red blood cells, and the heme moiety, the enzyme guanyl cyclase, which is found in the vascular smooth muscle cells and most other cells of the body. So therefore, when nitric oxide is formed by the vascular endothelium, it rapidly diffuses into the blood where it binds to hemoglobin and subsequently broken down. So it's difficult to measure. That's part of the problem is how do we measure nitric oxide itself in the blood, and how do we measure peroxynitrite in the blood? It's a tricky thing because it's such a short-lived molecule. Just a half-life of a few seconds and it's gone. So some of the cofactors, again, that is required for the production of nitric oxide, L-arginine in the body, which we kind of looked at. The second one, tetrahydrobiopterin, I want to look at in a little more detail because this is how it fits into the methylation cycles that we have covered in the past. Oxygen is required, and NADPH, which is produced in the citric acid cycle. And these factors will allow for the production of citrulline and then on to nitric oxide. Important component of the nitric oxide synthase is tetrahydrobiopterin, also known as BH4. This important molecule not only is important to make nitric oxide, but it's also critical to synthesize neurotransmitters. BH4 is synthesized in response to methylation. In fact, the enzyme that generates BH4 is known as the dihydrofolate reductase enzyme, or DHFR, and you can actually have a variant in that enzyme as well. This enzyme is activated when there's proper folate methylation. In other words, deficiencies of 5-methylfolate, either caused by the MTHFR gene mutation or some other factor that depletes folate, can lead to this BH4 deficiency, which leads to a nitric oxide deficiency. So again, here we look at inflammation and oxidative stress here, which will cause BH4 to be activated. And BH4 is also involved in the production of tyrosine, from phenylalanine to tyrosine. This is an enzyme, the phenylalanine hydroxylase enzyme, that's activated by BH4. Or BH4. Tyrosine hydroxylase, which converts tyrosine to DOPA, is activated by BH4. Tryptophan hydroxylase, which converts tryptophan to 5-HTP, is upregulated by BH4. And then nitric oxide will take arginine and make nitric oxide out of it through this enzyme, nitric oxide synthase. So you can see how critical this molecule is in this whole pathway. And it's all involving this folic acid, L-methylfolate, and SAMe. And we know that SAMe is our major methyl donor. It's what we're trying to make through the methylation pathway. We have to have SAMe in order to make BH4. So you can see how this is all tying in together with where we've gone before in the methylation pathway. So low BH4 then equals trouble. In addition to impairing the endothelial nitric oxide mediated NO formation, BH4 depletion may have additional detrimental effects in post-ischemic hearts. So after a heart attack, if you don't have enough BH4, then it's going to cause increased damage. So BH4 depletion not only prevents nitric oxide formation from endothelial nitric oxide synthase, but also results in markedly enhanced superoxide formation. And that, again, remember, that's our bad guy, our free radical molecule. So thus, BH4 has an important role in regulating endothelial nitric oxide synthase function, because BH4 depletion can switch the enzyme from the synthesis of nitric oxide to superoxide. So the role of BH4 in this enzymatic process is so critical that some research points to a deficiency of BH4 and thus of nitric oxide as being a core cause of the neurovascular dysfunction. That's the hallmark of circulation-related diseases such as diabetes. And don't forget that the Krebs cycle has to be running fully and regulated in order to make guanosine triphosphate, which is needed as its precursor to the BH4. So how is it impaired? How is BH4 impaired? Ammonia, if we make too much ammonia. So BH4 is critical in processing ammonia, which is a byproduct of the urea cycle. High ammonia will deplete BH4. Excess ammonia may occur in chronic bacterial or viral infections. High protein diets can do it. And then excess ammonia can occur with that CBS mutation that we've talked about in the past. If you have a CBS mutation, that pathway is going to be running a little too fast, and it can create a buildup of ammonia, which will deplete the BH4 and therefore cause some superoxide molecules to be created and will lower your nitric oxide. Vitamin D, we talked about last webinar on the vitamin D receptor, and it also plays into this whole pathway. So vitamin D could play a vital role in the regulation of cardiovascular function and in the risk of several disease states by controlling arterial stiffness and levels of nitric oxide, according to new research in mice. And vitamin D status is important in the regulation of nitric oxide production, and then the VDR, or the vitamin D receptor variants, will interfere with nitric oxide production. So I think maybe you're getting the idea, and I hope I'm getting through that you've got to look at all these different variants along this pathway and make sure that they're all running smoothly. And that's what we're doing we're teaching these seminars all around the country. We've got many locations throughout the country this year, but we're teaching you how to put this all together. And I know this is an overwhelming amount of information, but I really come back to the kinesiology. It's what helps me sort this out. So last time we talked about, you can test the vitamin D receptor by putting vitamin D on somebody, and if it goes weak, then we know that that person does not absorb vitamin D efficiently, and they may have a VDR defect in their genetic makeup. So we added vitamin K and vitamin A into the program, and vitamin D then will test strong, and it will begin the production of vitamin D needed to make this next pathway run, which is the nitric oxide synthase pathway. But it also requires the methylfolate and the B12 in the original pathways to get that going too, and then normal production of the ACAT pathway, which feeds into this as well. You can also, I looked online and I just ordered these because I wanted to play with them. I haven't played with them yet, but there are different places where you can get nitric oxide indicators that you can use, salivary. So it helps you kind of monitor what's going on in your body if you're creating not enough nitric oxide or maybe even too much. You can measure it on these sticks and they're fairly inexpensive. So I'm looking forward to playing with those. I just went on Amazon to find it, and there are different brands. I didn't want to put a brand name here because I just don't want to advertise any one brand, but I thought I got a couple different kinds just to try them out, so I'll keep you posted on that. So here's the bad news, the peroxide production from nitric oxide. So what happens is several hours after exposure to cytokines or endotoxins, macrophages, or white blood cells, and neutrophils begin to produce large quantities of nitric oxide, and they continue to do so for many hours. They're just pelting these infections with, just that's our ammunition to kill off the bad guys. They also produce O2, which is a superoxide anion, which chemically combines with nitric oxide to form an even more toxic peroxynitrite molecule. That's the O-no molecule, which rapidly reacts with water to yield a highly reactive hydroxyl radical and NO2. These are used to kill bacteria. So there we are right there. So there's the nitric oxide plus the superoxide molecule, and that results in peroxynitrite. If you have adequate amounts of superoxide dismutase, then you're just going to make hydrogen peroxide, and then that's broken down by catalase into water and oxygen. So when you cut yourself and you pour hydrogen peroxide on a cut and it starts to bubble up, well, that's the action of catalase on your skin, and what it's doing is it's breaking up the enzymes into hydrogen peroxide, the hydrogen peroxide is breaking up into water and oxygen. It's the release of that oxygen that's bubbling up on your skin. So if you have a variant in this superoxide dismutase enzyme right here, and you don't make enough superoxide dismutase to quench the superoxide molecule, then you can see how this can lead to excess production of peroxynitrite, and that's where we get into trouble. And that's the O-no factor. Interesting thing. So if we take a look at how we get nitric oxide into the body, it's just kind of fascinating. I learned this just through studying. But we have the nitrate molecule that's converted into the nitrite molecule in the mouth. It's in the oral cavity, and that's done by the action of bacteria here. And then in the stomach, that nitrite molecule is converted to nitric oxide in the presence of stomach acid. And that helps regulate gastric function. Remember, we talked about peristalsis action, but it also helps the gallbladder function normally. So over here, this is the nitric oxide pathways that we've kind of talked about, but the fascinating thing is that chronic use of antibiotics or mouthwash, in our efforts to have a sterile mouth, we're really kind of doing ourselves a disservice. It can create problems absorbing nitric oxide, the conversion of nitrates to nitrites from food, which occurs in the mouth. And likewise, antacids will inhibit the conversion of nitrites to nitric oxide that occurs in the stomach. So we can really kind of mess ourselves up. I go into patients' homes here, and I have one particular patient that when I go and look at right beside their sink is a bottle of the antiseptic hand wash, which they use. It's by every sink, in their bathrooms, in their kitchen, and oh my gosh, and in the bathroom there's big old bottles of mouthwash there, and I'm like, oh my gosh, we're so germophobic that we're really killing off all our good bacteria, and it's really doing us a disservice. And this was an interesting little piece of the puzzle here where mouthwash can really kind of create some problems with creating nitric. Who knew that mouthwash could lead to some cardiovascular issues? So there you go. Throw it in the trash. Low nitric oxide can also occur because of a lack of it in the diet, and veggies and meats. The veggies are going to contain directly, they're going to contain nitrites and nitrates, and meats are going to contain arginine. So, and low activity of the nitric oxide synthase enzyme due to genetic variants, and I'll show you those in a little bit. Aseptic oral cavity through mouthwash, an aseptic gut through low probiotics and antibiotics, and low BH4 production from methylation defects, or lack of substrates, which are arginine, citrulline, B12, folic acid, again, the methylation products, and low vitamin D absorption through vitamin D defects or lack of sun exposure. And that's another way we can get nitric oxide is through sun exposure, so created in our skin. So high arginine foods are there, the nuts, peanuts, etc. So in our diet, I want you to get sort of this little tidbit as well. So in our diet, our proteins are converted to arginine. And this pathway over here is so here's the nitric oxide synthase gene, or enzyme that's created by the gene coding for it. So arginine is converted to nitric oxide and to citrulline. And from there, we have a gene right here, this lovely little ASS gene, which converts citrulline back to arginine. So it's kind of a self-contained unit over here, and if you don't make nitric oxide synthase, or if you're making it inefficiently, let's say, then arginine is going to go over here and create creatine, or it's going to create urea, or it's going to create ornithine over here and possibly go to glutamate. You can also have defects in the OTC gene, which will prohibit ornithine being made back into citrulline. So you can see how, so there are several areas where you can have genetic variants that can block these pathway anywhere along the road here. But this one, it tends to be a little more common than some of the others. And if you're making that, then taking arginine is probably not a good idea, and you can see why. So if you're inhibited here, then arginine is going to go ahead and go this direction. So arginine can be a good thing in creating nitric oxide. However, if you have this variant here, then arginine probably is not going to be the best way to do that. Alrighty. So independent of the nitric oxide synthase pathway, the nitrate, nitrite, nitric oxide pathway increases nitric oxide from green leafy vegetables, especially spinach and arugula and beets. And the cool thing about beets especially has all the antioxidants in it, along with the nitrites and the nitrates. So beets will clean up the superoxide molecule and increase nitric oxide all at the same time. So these foods increase cardioprotective levels of NO and help reduce blood pressure in pre-hypertensive persons. The reduction of nitrites occurs in the mouth by bacteria, like we talked about, and they can be measured in the saliva. And there was a question about what are the sticks called, and they're just nitric oxide. What did I Googled nitric oxide in the saliva testers. So you can just Google that or go on Amazon and look up nitric oxide testers, and you'll probably find them there. I think they go by different names, but start there, and that'll should bring up something for you. So beetroot, one of the best ways that you can get nitric oxide in the diet. And vegetable juices, carrots, pomegranate, red wine, green tea, and acai contain nitric oxide promoting ingredients. So here's a look at some of the other vegetables. What we want are the ones that have the antioxidants here in the ORAC index, and also have nitrites and nitrates. So the highest being kale, Swiss chard, arugula, spinach, have both in spades. So it's another good reason to eat your vegetables. So it's interesting when you compare a Western menu with a Mediterranean menu, and you can look at the difference here. So the nitric oxide levels here in the Western menu, a little tiny bit, and then the Mediterranean menu has lots of nitric oxide. So for breakfast, they had a bagel with cream cheese and black coffee, carrot nut muffin, and a Diet Coke, Big Mac, large French fries, Diet Coke for lunch, black coffee for a PM snack, a cheese pizza, and Diet Coke. I don't know who does that. It just makes me cringe. Does anybody drink that stuff anymore? I just don't know. But the Mediterranean menu, toast with jam and butter, and they had a cappuccino, a yogurt, and a carrot juice for an AM snack, Mediterranean wrap and garden vegetable soup and mineral water, trail mix, orange juice, salmon, and red wine, much higher in nitric oxide with these minor changes. So I thought that was kind of fascinating. So other ways that you can increase nitric oxide, again, we talked about nitrite and nitrate-rich foods, grapeseed extract, vitamin C, garlic, citrulline, arginine. Exercise increases nitric oxide. Pycnogenols, which actually reduce superoxide and therefore increase nitric oxide that way. Sunlight, we talked about, resveratrol. Ginseng, quercetin, CoQ10, don't use your mouthwash. Laughter increases nitric oxide. Niacin, capsaicin, coffee, raw cacao, and horny goat weed, and essential fatty acids all increase nitric oxide production. So, but when we get the excess nitric oxide, remember, too much of a good thing can be a bad thing. And when it turns to the peroxynitrite molecules, it compromises the blood-brain barrier and leads to leaky gut. It inhibits the gallbladder contraction and causes the retention of bile. This can be one of the reasons for constipation. If you've got some patients who are just unresponsive in the constipation department, it can be from this peroxynitrite molecule being created. Also causes neurodegeneration and demyelination and breaks down the protective lining of the gut, or the mucin. Causes blood vessel constriction. It's a major free radical and it destroys nitric oxide itself, cross-links the proteins, and causes damage to DNA. So in some studies, recent evidence indicates that most of the cytotoxicity attributed to nitric oxide is rather due to peroxynitrite produced from the diffusion-controlled reaction between nitric oxide and another free radical, the superoxide anion. So peroxynitrite interacts with lipids, DNA, and proteins via direct oxidative reactions or via indirect radical-mediated mechanisms. These reactions trigger cellular responses ranging from subtle modulations of cell signaling to overwhelming oxidative injury, committing cells to necrosis or apoptosis. In vivo, peroxynitrite generation represents a crucial pathogenic mechanism in conditions such as stroke, myocardial infarct, chronic heart failure, diabetes, circulatory shock, chronic inflammatory diseases, and neurodegenerative disorders. So one of the other activating pathways looks like this, just to put it down simply. The NMDA receptor, that's the glutamate receptor. So high glutamate levels can also turn this pathway on. The channels allow calcium to enter into the cell, which causes it to depolarize. And that depolarization causes neuronal nitric oxide synthase and endothelial nitric oxide synthase activation, which increases nitric oxide, and then it reacts with superoxide to form the peroxynitrites. So, again, this is kind of a reiteration, but it causes all kinds of problems. Prostaglandins increase, that's inflammation. Decreased response to glucocorticoids, so issues with adrenal problems. Increased, prostaglandin production. Glutamate transporters, glutamine synthesis, tyrosine hydroxylase, it causes interference with that enzyme and decreases norepinephrine formation, causing more adrenal problems and reactive oxygen species. So here are a couple of studies relating it to blood pressure issues. Accumulating evidence suggests that alterations in nitric oxide synthesis and the reduction in the bioavailability of endothelium-derived nitric oxide by increased oxidative stress are key contributors to the pathogenesis of hypertension. Numerous experimental studies using animal models of disease and clinical reports have established that hypertension is associated with increased superoxide production, dysregulation of nitric oxide synthase, and endothelial dysfunction. So, again, we're looking at hypertension, obesity, dyslipidemias, particularly hypercholesterolemia and hypertriglyceridemia, diabetes type 1 and 2, heart failure, atherosclerosis, and aging. And it's an interesting, I did this huge study of one patient, but it was my mom. She, I talk about how I'm using her as a guinea pig because she's got some interesting things going on, and she had hypertension. She's had it since I was born, so 50-some years here we're talking about she's had it, and tending to get a little mid-center obesity going on, and definitely dyslipidemia. She had cholesterol over 300 for most of her life and high triglycerides to the point the doctors are saying, statins, statins, statins. And I'm saying, no, 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 do not. You know, I'll take care of this. Tending toward diabetes, definitely. Her hemoglobin A1C was going up. And heart failure, I don't know. She didn't really have any symptoms of that except maybe a little bit of wheezing going on where you could hear some accumulation in the lungs. And atherosclerosis, we had her calcium scores done, and there was definitely a little bit of that going on, and aging. So I put her on a program, and I have her case study toward the end of this presentation that I wanted to share with you, and how it dropped it dropped her blood pressure, it dropped her cholesterol, it dropped her triglycerides, and it dropped her hemoglobin A1C considerably. Cholesterol over 100 points, and her hemoglobin A1C went from 6.2 down to 5.6. Doing nothing, no change in diet, only the methylation, and we put her on a nitric oxide enhancing program, and bam, it just dropped. It was stunning, and the doctor was, I took her back to her doctor, and they were just blown away. So let's talk about angiotensin II and the nitric oxide synthase enzymes. It's a well-known factor in the pathogenesis of most cardiovascular disorders and a natural regulator of blood pressure. What it does is it induces superoxide formation, and we now know that that combines with nitric oxide and lowers nitric oxide, takes it out of the circulation. Recent studies have demonstrated that angiotensin II can induce peroxynitrate formation, and angiotensin II increases NADPH oxidase, which produces superoxide. This in turn increases peroxynitrate and depletion of BH4. So let me show you that pathway here. So remember when we talked about this upregulation of a CBS, if you've got a variant here, a genetic variant, you're going to tend to go down this pathway faster than normal, and things can back up on you. And remember we talked about you can create more ammonia, and that depletes your nitric oxide right there, and it's going to create a deficiency of glutathione, which again, this is one of our major antioxidant molecules, so we need that to scoop up sort of the free radicals, and if we're not making that, that's another insult. But then if you look down here, it causes the buildup of sulfites, and it goes down this pathway to increase the production of angiotensin I to angiotensin II. It's inhibited by nitric oxide, but it also inhibits nitric oxide, so it's a negative feedback loop. And this leads to high blood pressure and cardiovascular disease. Low cortisol has been shown to increase susceptibility to chronic fatigue syndrome and myalgic encephalitis. This may be due to the role that cortisol plays in lowering inducible nitric oxide synthase induction, or low cortisol, which increases nitric oxide levels. Its involvement in irritable bowel syndrome is fascinating. Increased expression or activity of inducible nitric oxide synthase, as well as elevated levels of luminal nitrite, have been detected in patients suffering from ulcerative colitis and Crohn's disease. And I have a patient who had, since she was 12 years old, she's had ulcerative colitis and has been in and out of the hospital for flare-ups with that. And on one methylation program, it just stopped. It just completely stopped. It was just stunning. She's my biggest proponent now, but now I see I didn't really quite understand the mechanism until I started looking into this pathway a little deeper, and it looks like it just turned off the production of the wrong kinds of nitric oxide here. And peroxynitrite generation has been shown both in experimental models of colitis and in human samples of active Crohn's disease lesions. Peroxynitrite formation has been implicated in Alzheimer's, Parkinson's, Huntington's, MS, ALS, and traumatic brain injury. So again, we have, here's the peroxynitrite, nitrites. I keep saying nitrates. Just interpret that as nitrite. And it leads to neuronal damage here, microglial activation. So it's this superoxide molecule here and low BH4. So we run out of BH4 up in the pathway here, and it leads to all this destruction. If you've got a genetic variant here in the SOD enzyme, then you're going to have higher levels of hydrogen peroxide. Sorry, you're going to have lower levels of hydrogen peroxide and higher levels of superoxide and peroxynitrate. It's just going to block this. If you're adequate in your superoxide dismutase, then it's going to drop these superoxide molecules down into hydrogen peroxide. This catalase enzyme then breaks it down into water and oxygen molecules. So again, you can have variants in each of these enzymes that will keep this from happening and create a buildup of these oxide molecules over here. The pathogenic role of peroxynitrite in traumatic brain injury is supported by evidence demonstrating increased brain 3-NT levels, that's nitric, that's peroxynitrite, following traumatic brain injury in experimental mouse and rat models, and by the beneficial effects of nitric oxide synthase inhibitors and peroxynitrite scavengers in reducing neuronal injury and improving neurological recovery following injury. I thought that was an interesting little tidbit there. So this was an interesting, I kind of grabbed this from another lecturer, this slide, but I thought it was an interesting few studies here on sauna and how it raises tetrahydrobiopterin and therefore increases nitric oxide. Because NF-kappa B controls many genes involved in inflammation, it's not surprising that NF-kappa B is found to be chronically active in many inflammatory diseases, such as inflammatory bowel disease, arthritis, sepsis, gastritis, asthma, atherosclerosis, and others. Incorrect regulation of the NF-kappa B has been linked to cancer, inflammatory and autoimmune diseases, septic shock, viral infections, and improper immune development. NF-kappa B has also been implicated in the process of synaptic plasticity and memory issues. So basically, we have an inactive NF-kappa B molecule there, and it's activated by a reactive oxygen species here, free radicals, turns on kinases, and then activates the NF-kappa B right here, causes the tumor necrosis factor alpha to come on the scene, and then cause a respiratory burst of inducible nitric oxide, and then they turn into the reactive nitrogen species, which is our peroxynitrite. White blood cells use nitric oxide not only to kill infectious agents such as bacteria, fungi, and parasites, but also to defend the host against tumors. So it's an interesting thing. Studies are being done on whether nitric oxide can be used to stop the growth of tumors, since nitric oxide induces apoptosis. And there's been some association with MS. Direct evidence includes the fact that inducible nitric oxide synthase mRNA has been identified in multiple sclerosis plaques, with evidence that macrophages, astrocytes, and microglia within active lesions express high levels of inducible nitric oxide and endothelial nitric oxide. There's also evidence for the presence of reactive nitrogen species in MS through nitrotyrosine, a marker of the presence of peroxynitrite, which is found in the diseased brain, particularly in areas of demyelination and inflammation. So what causes this uncoupling? And what I mean by uncoupling is the conversion of nitric oxide to peroxynitrite. Smoking, inactivity can do it, not enough exercise, too much arginine or too little arginine, oxidative stress, low oxygen, high blood glucose, and low blood levels of BH4, as we talked about. The interesting thing, bacteria also, its bacterial nitric oxide synthase has been shown to protect bacteria against oxidative stress, diverse antibiotics, and host immune response. So they make their own nitric oxide. It's kind of interesting. But bacterial nitric oxide synthase plays a key role in the transcription of SOD, and it helps shield the bacteria against conventional antimicrobials. So we're going to talk about BH4 in a minute here, but the way that we increase BH4 is up and coming. It's an incredibly important molecule. Nitric oxide is a very important molecule in the regulation of cerebral and extracerebral cranial blood flow and arterial diameters. It's also involved in nociceptive processing. So it's involved in migraines. Blockade of nitric oxide synthase effectively treats attacks of migraine without aura. Similar results have been obtained for chronic tension type headache and cluster headache. Nitric oxide may play a key role in migraine and other vascular headaches, since glycerol trinitrate, it's a donor of nitric oxide, and histamine, which probably activates endothelial nitric oxide formation, both cause a pulsing dose-dependent headache with several migrainous characteristics. In other words, they could induce a migraine by increasing histamine and nitric oxide. Inhibition of the breakdown of cyclic GMP also provokes migraine in sufferers, indicating that GCMP is the effector of nitric oxide-induced migraine. A migraine may be caused by increased amounts or affinity of an enzyme, and the NO-triggered cascade of reactions may be involved in the pathogenesis of other vascular headaches. Too much or too little, or the wrong kinds. Okay. I'm going to go past this. I just did so much information. I got kind of carried away here, but in the interest of saving time, because I want to get into some more interesting information, these slides will be available when we have the webinar up on the websites. It's on my website as well, and I'll give you that information in a minute. But basically, I did find a huge correlation between migraines and nitric oxide or peroxynitrites. There was some association with autoimmunity, and here's some studies. And demyelination, for sure, is definitely has been shown with excess peroxynitrite formation. There's more demyelination studies, MS studies, MS studies. Interesting little tidbit here. Peroxynitrite damages the myelin sheath through lipid peroxidation. The causative role in peroxynitrite in oligodendritic cell damage is supported by findings that a peroxynitrite scavenger, uric acid, can inhibit demyelination and axonal damage. So note that gout and MS are mutually exclusive conditions. So uric acid is one of our major antioxidants in the blood. So it's a fascinating little tidbit that lowering our nitric acid, or sorry, our uric acid through uric acid inhibitors like what's the medication? Allopurinol can cause an increase in peroxynitrites. So I thought that was a fascinating little tidbit there. And Lyme, that found some interesting research. High serum levels of nitric oxide, MDA, and nitrotyrosine observed in patients with Lyme borreliosis indicate an enhancement of lipid peroxidation and protein nitration, which may enhance the inflammatory process in patients with Lyme. Mitochondrial superoxide levels were significantly higher in Lyme borreliosis patients as compared to healthy controls. So we have a higher production of peroxynitrites associated with Lyme. Human skin exposed to solar ultraviolet radiation on a daily basis, it's generally accepted that mutagenic and carcinogenic properties of UVB is due to the direct interaction with DNA. On the other hand, UVA induces the formation of reactive oxygen species, which plays a pivotal role as mediators of UVA-induced injuries in human skin. UVA-induced decomposition of the nitric oxide, that should be S, like nitrite, leads not only to non-enzymatic formation of nitric oxide, but also toxic reaction nitric species like peroxynitrite. Whereas under anti-oxidative conditions, the generation of protective amounts of nitric oxide is favored. Under oxidative conditions, less injurious reactive nitrogen oxide species are generated, which may enhance UVA-induced cell death. So basically what they're saying is if you have high levels of reactive oxygen species like superoxide and peroxynitrites, and you're exposed to UVA, it's going to make it worse. But if your pathway is working correctly, then you're actually going to increase nitric oxide and have no adverse effects from the UVA and UVB exposure. It's been suggested that many of the beneficial effects of sunlight, particularly those related to cardiovascular health, are mediated by mechanisms that are independent of vitamin D production. It's also been hypothesized that photolabile nitric oxide-related species or compounds such as nitrite and nitrosothiols, which are stored in comparably high concentrations in the skin, can be mobilized by UVA and delivered to the systemic circulation, exerting coronary vasodilation and antihypertensive effects. So sunlight, sun exposure, can induce vasodilation and lower your blood pressure. However, despite the controversial discussion of the beneficial and hazardous effects of sunlight, there's incontrovertible evidence that UVB and UVA can induce cell damage, which in turn causes necrosis, apoptosis, and autophagy. So basically, again, just repeating what we just said. I'm going to go past this a little bit. Same thing, skin protection. So again, here UVA is inducing cell survival when you have high levels of antioxidants, and cell death if you have low levels of antioxidants. Okay, given that expression of the main glutamate transporters and oligodendrocytes is suppressed in MS and It's possible that NO helps to perpetuate the glutamate-mediated damage to oligodendrocytes and neurons during inflammation by both increasing the release of glutamate and suppressing its reuptake. So we need, glutamate we looked at as in the GAD variant. If you have a GAD variant, then you're not going to break glutamate down to GABA as easily, and you're going to have higher than normal levels of glutamate, which then induces the nitric oxide pathway and peroxynitrites. And that's the microglial activation pathway there. That's actually in my interactive chart. Here's when we look at the MTHFR support report. Here are the nitric oxide synthase genes, and in this person, there's lots of. When we look at the green over here, this means it's normal. That enzyme is functioning normally. When we look at a yellow, that's a heterozygous variant, and it can be reduced by about 30%. And so this person has not bad. Nitric oxide pathway looks not too bad here. Maybe a little bit of a deficiency, but pretty minor when you look at the overall total of the enzymes. These nitric oxide synthase variants, this NOS2 specifically, is associated with multiple sclerosis, so cerebral malaria, Alzheimer's, and Lewy body dementia. This gene encodes nitric oxide synthase, which is expressed in the liver and is inducible by a combination of lipopolysaccharides and certain cytokines. Borderline leprosy and bladder carcinoma in situ are associated with that variant. The NOS3 variant is going to be associated with this is more the cardiovascular kind of stuff, thrombosis and stroke, essential hypertension, glaucoma, migraines, Alzheimer's, Lewy body dementia, and eclampsia and preeclampsia issues. And so there are different tests. One is an exhaled nitric oxygen test that can help with diagnosis and treatment of asthma. It measures the level of nitric oxide gas in an exhaled sample of your breath. This sample is collected by having you breathe into a mouthpiece of a machine that then performs a measurement, or you can get the nitric oxide indicator strips through various companies. And when we already looked at the citrulline versus arginine pathway, and again, once more, if you've got a nitric oxide synthase variant, arginine may not be the best thing for you to take, but if you don't, then arginine can produce nitric oxide. Let's go on forward here. Just wanted to point out how the MDs treat this kind of thing, nitric oxide pathway issues. And if you look here, remember we talked about angiotensin II causing a decrease in nitric oxide. Well, they can give you an ACE inhibitor to keep that from happening, and that lowers your blood pressure. There are these blockers here that can block the production from angiotensin II to nitric oxide. And arginase inhibitors, L-arginine and citrulline, can help. They block this arginine pathway here. ADMA inhibitors can block it over here. Statins, polyphenols, etc., block this pathway here in the production of endothelial nitric oxide. Here's BH4 there. You can have organic nitrates, or nitric oxide inhalation can increase this pathway down here. Organic nitrates and nitrites enter this way. These stimulators will increase production of nitric oxide here. Those PDS inhibitors, PDES, this is Viagra, and this is how it works by keeping nitric oxide from breaking down. So GMP will break down nitric oxide, so it blocks that enzyme here. So how do we increase nitric oxide? We balance methylation. We increase nitric oxide in the diet or supplementation. And we decrease the O-no production pathway. And we reduce stress on the methylation pathway, optimize antioxidant production, and supplementation. Oops, sorry. We also increase BH4 through methylation, so let's talk about that for just a moment. We also increase citrulline, increase the production of NADH and NADPH. I'm going to jump ahead here. Let's look at, I have a pathway on BH4 here. Okay, let me go back. So to challenge those of you that are kinesiologists and muscle testers, we have in your kit, if you have one of the kits, we have nitric oxide, superoxide, we have hydrogen peroxide, we have ornithine, arginine, and citrulline. And those can all help to challenge this pathway. We're also going to be adding peroxynitrites, NF-kappa B, inducible nitric oxide synthase, and hydroxyl radicals. So you can take a closer look at what's going on here in this pathway. So some of the supplement supports here, we have total arginine, which is great if there's no nitric oxide synthase variant present. Definitely that superoxide product will help clean up some of the, misspelled antioxidants, but it will help with antioxidants. VDR support, we have Complete K, we have vitamin A, we have Complete High D3 to increase the vitamin D. We have methylation balance, B complex, which includes the active forms now. The new B complex with NutriWest has the methylfolate and the methylcobalamin in it. Then we have the 5-MTH folate, hydroxy or methylcobalamin. And then the comp support. When you get all those balanced, then the nitric oxide pathway is more likely to stay on the right side of the pathway. We also want to do ACAT support, so citric acid cycle factors can come in with that, CoQ10+, biostress B, helps the thiamine in that pathway, helps the citric acid cycle work correctly. Total alpha lipoic acid and L-carnitine will help upregulate that pathway, and it'll decrease your loss of nitric oxide and increase your production of the right kinds. Again, increasing BH4, I want to talk about that again in a minute here. Increase NADPH and GTP production through increasing Krebs cycle function through carnitine, CoQ10, and ribose, and then increasing antioxidants. So there's a list of some of the things that can really help. Superox, sorry, I said it wrong. I said superoxide rather than superox. It cleans up superoxide molecules. Okay. Going past this one, use both. We know about superoxide. So I'm going to go past this. There's a lot in here about the superoxide molecule, but we're running out of time, and I want to say some other things before we end. But basically, you can have a variant, a genetic variant in the SOD gene that will cause an increase in reactive oxygen species, which are the hydroxyl radicals and superoxide anions and peroxynitrites. Mutations. Okay. So here we see the SOD3 down here. This person has a double variant, and so they're not going to make superoxide dismutase correctly, and therefore they're going to have higher levels of superoxide, and they would highly benefit from the superox supplement. It's interesting that levels of SOD can vary as much as 50% in individuals. This may explain why some people age quickly and others live happy, healthy, long lives. And research conducted in the early 1980s by Richard Cutler at the Gerontology Research Center at the NIH, mammals that produced higher tissue and serum levels of SOD lived longer than those with lower SOD levels, so it's a really big factor in aging. So there's the different variants that you can have and the different types of SODs. There's 1, 2, and 3. So I'll let you look at those when you go back in to look at the at this webinar. You can take a closer look at those slides. Interesting, they identified 13 mutations in the SOD gene in ALS patients. So that's another thing to look at if you have ALS patients. Multiple chemical sensitivity it's associated with too. And then there are genetic variants in the CAT gene, and this is the catalase that breaks down hydrogen peroxide. And in the Lyme, this is a Lyme case study where we had an 11-year-old with a history of extreme pain for 3 years, wheelchair bound due to pain, excessive food allergies, leaky gut, especially gluten and dairy intolerance. 40 doctors were consulted, all tests were normal, a diagnosis of reflex sympathetic dystrophy. History of tick bite and bullseye rash, and lived near Lyme, Connecticut, and we did find Lyme on him and began to work with him, and all I did was a methylation pathway and just brought him out of all of these issues very quickly. The food allergies went, the leaky gut went very quickly. Within 2 to 3 weeks, it was gone, and the pain went. So it was just a fascinating study here. And then again, this is in my practice, and it's not a huge study, but I think it's an impetus when we see these kind of things for a larger study to look at this whole what's going on in this entire methylation pathway and how powerful it is. And these were his genes. We look at the MAO, MTHFR, and MTR, and we can see that he wasn't even methylating in the first place. This right here. So if he's got a genetic variant here, he doesn't absorb folic acid, and this one will keep him from absorbing B12. Both are which critical in making the methyl groups needed to get the nitric oxide synthase pathway running. He also had, when we looked at his larger study in the MTHFR support, he had an SOD gene, which again keeps him from breaking down superoxide, so high levels of superoxide molecule. And he had the CAT gene right there, so it keeps him from breaking down hydrogen peroxide. He also had the GAD gene. He had a couple of variants there, which keep him from breaking down glutamate. So these three, along with not even making methyl groups in the first place, really set him up for this whole situation. And this is, again, my mother, which I told you just a little bit about, and she had BDR, MTR genes, nothing major in her genetic genie, but she did have all these MTRRs, which keep her from making BH4. So it's low B12 and low folic acid, the primary methylation generators that make BH4. And we'll show you in the pathway in just a moment, again, what that is. She has, you can see, issues in the nitric oxide synthase pathways right here. 2 big homozygous variants where she doesn't make nitric oxide synthase. So she didn't even make it in the first place. So using arginine and citrulline and some other nutrients really helped her make nitric oxide. These were her supplements. And here's her cholesterol. So we started on 2010, and you can see her cholesterol was running 300s. We measured it every year. And then, boom, once we started on the methylation program, we did nothing to just directly hit her cholesterol. It dropped significantly. LDLs all the way down to 137 from 253, the latest high. Triglycerides from 156 down to 75. Her LDLs dropped too, which this is a little bit lower than I like to see it, so we're going to be working on that. But her hemoglobin A1C dropped from its highest at 6.2 down to 5.7. Pretty stunning results. And again, we didn't do a statin. We didn't even do a natural statin or red yeast rice or anything like that, just methylation. And it had this tremendously powerful effect on her cardiovascular health. So here I'm going to be all over the country. We do a whole weekend where we go through a lot of detail through the methylation pathway. Some areas I want to point out to you. We already did the San Diego one in Dallas. These are the beginning ones unless it says advanced. So those of you who have taken the methylation pathway classes in the past, we are going to do an advanced one in Dallas in April, and we're going to be doing Denver down here in August. Actually, this starts on the 26th. We're doing the whole 2.5-day one in Denver, Colorado. So Dr. Tui is excited to get us into Denver finally for those of you in that area. Very excited to go to her area of the country. And then if you're wanting to learn the holistic kinesiology, we have classes starting in Phoenix in February and March. This will be part 1 and part 2. So those of you who don't do kinesiology, I really highly recommend it. It's very difficult. You can do it. You can do the analysis of the methylation pathways through blood work and saliva, etc., but it's pretty hard to really get it balanced. It's so much easier to see what's going on with the kinesiology, and we'll teach you how to do that in those seminars. And just a reminder, here's our test kit. We use these little testers to see what's going on in the pathway and challenge these pathways to find out what's going on. And we'll be adding a few to it now that I know a little bit more about the nitric oxide pathways. You can see here's a superoxide molecule right there. And so there are ways to kind of evaluate what's going on, but we're going to get into it a little bit deeper. This is the poster that we designed, and these are the challenges here. If you come up with a challenge, it tells you what SNP, or single nucleotide polymorphism, you're likely looking at if you have a weakness in these challenges in that kit we just showed you. And then you go over here and you look up what supplements that you would need to take in order to balance that particular SNP. So it's a handy little poster. And NutriWest has created a methylation test kit for us, and it's full of all the nutrients you need to get this pathway balanced. It's an amazing little handy tool to have. And once again, so if you take a look, I wanted to show you the BH4 pathway. So there were some questions about that, and here it is right there. So again, this is folic acid, B12 pathways. These have to be going in order to make this BH4 molecule right here. This intersects with the folate pathway, so it's critical. You can have a genetic variant in here too, but you need this pathway running in order to have this nitric oxide synthase pathway running too. That's why they're butting up against each other. So critical. This is what we covered in the first webinar. This is what we covered in the second webinar. And this is what we covered in the third webinar. And this is the fourth here. The fifth one, we're going to be looking at these whole pathways here, the histamine pathway, and then the citric acid cycle pathway and its effect on methylation. So you can get those products at www.holisticmethylation.com, in addition to the interactive chart, which is a great learning tool for learning the methylation pathways. And there's my email, holisticmethylation@gmail.com, and contact your NutriWest dealer in your area for more information about the test kits and upcoming seminars. And that number again is 1-800-443-3333 for contacting your NutriWest distributor. So basically, that's it. I'm sorry to go over time. There was just so much information, I couldn't help myself. So are there any questions that we have before
Yes, as a matter of fact, that was fascinating, Jay. Thank you so much. One of the questions here is whether or not you periodically use the indicator strips on patients to prove that the supplementation protocols are working.
Well, I haven't yet. I just found out about them, so I ordered them, and I've got 2 of them coming in. And like I say, I'll keep you guys posted on it. I'm interested to see if we can tell, you know, when we take certain nutrients, will it increase the nitric oxide in the saliva or the nitrate level in the saliva? So it'll be interesting.
Great. Okay. Well, we had a lot of nice compliments come in, and I would like to add one. That was really interesting. So thank you again, Dr. J. This is just wonderful, especially to have 4 in this series and be able to refer to the website for more information. So when we have the date for the next in the series, we will shoot out an email and let everyone know. And until then, thank you everyone for showing your support and joining us in the webinar. So thank you, Dr. Dunn.
Thank you, Dr. Tui. I really appreciate it.